Alright, this is the overdose, section 1.2. We're going to talk about molecular transport. Okay, we said we have different kinds of body fluid, and we said that those body fluids have different concentrations of various molecules. Now, some of those molecules have to move across the membrane. So we have particles out here in our blood and in our interstitial fluid, and we have these cells, and these particles need to get into the cells in order to be metabolized, in order to be used, in order to be turned into various things. So we'll just call this moving into the cell. Okay, so we have molecules in the ECF, and they're going to interact with a membrane, and they need to get across that membrane. Now, some things can move across freely, other things can't, so the membrane is said to be selectively permeable. And we need to be able to get nutrients into the membrane and waste out of it. And there are two basic ways that molecules move past a membrane. The first way is called carrier mediated. The second way is called non-carrier mediated, if that's not hard to remember. And the non-carrier mediated is exemplified by simple diffusion. The carrier mediated, you have two things to think about. There's something called facilitated diffusion, and there's something called active transport. So let's go back and talk about simple diffusion again really quick. In simple diffusion, you have hydrophobic molecules that can move directly across the lipid membrane. So this is called lipid soluble. Now, some textbooks will classify channel proteins as also being simple diffusion. Others will say that it's facilitated diffusion. I'll explain that in a second. But channel proteins, and what I'm talking about here is if you have your lipid membrane, there can be a simple ion channel or some kind of channel. I'm going to cut it as if I've cut it in half so that you're seeing right down the middle of it. So you have this molecule and it's moving through this tube essentially. The tube is called a channel. Now, some textbooks will call this facilitated diffusion, others will call it simple diffusion, because the channel just exists. It doesn't move, it doesn't do anything, it doesn't behave in any way, it just exists. Regardless of what you call this channel protein, facilitated or simple diffusion, there are two major types of channel proteins. You have your water channels, which are aquaporins. With all channels, you can either have gated channels or non-gated. What that means is I could have a little chain sitting on here that has a ball attached to it, and that ball blocks the channel, and that whenever something comes and stimulates that thing to move, then the channel will be open. You can also have channels that close up and have a ball blocking them, so that the channel has to be opened and the ball has to be removed before the channel is free to allow diffusion. Now, as I implied, channels can be opened or closed, and so because channels can have activity, sometimes the channels are classified as being facilitated diffusion. And so with non-carrier mediated transport, we've talked about simple diffusion, where we can simply have a molecule move straight through a membrane, and we've also talked about possible channel proteins, and channel protein being basically just a tube. Now, with facilitated diffusion, it's going to behave the same way. So with diffusion, molecules always move from highest concentration to lowest concentration. So if I have a low concentration on this side and a high concentration on this side, it's going to move in this direction. Anything with the word diffusion in it is going to work the same way. So here where I have facilitated diffusion, the difference would be I've got my high concentration of solutes or molecules or whatever they happen to be. I have my membrane, and then I have this carrier that will grab a hold of, as soon as this diffuses into this area, it will cause the shape to change and the molecule will move into the lower concentration. Now let's talk about active transport. Active transport means that I have a low concentration gradient here, and I have a transporter, let's say a transporter, similar to the facilitated diffusion, and that transporter is going to require ATP. ATP gives this energy for the transporter to move, and whenever this thing gets close, it's going to grab it and push it through down here. So I'm going to say I have a high concentration already down here, I have a low concentration up here, but I'm going to use ATP to grab these things and push them into the higher concentration area. Now with all of these things that have proteins, so all of the carrier mediated, and possibly only the carrier mediated depending on how your textbook classifies it, but the carrier mediated, the active transport, the facilitated diffusion, and these channel proteins over here, all of these have a couple of very important things in common. So first is they all have specificity. Secondly, they all have competition. And thirdly, saturation. So let's create some more space and talk about those three things really quickly. The first one we said was specificity. This means that the protein is able to recognize specific compounds or molecules so that only those molecules can get through. Next, let's talk about saturation. We'll save competition for the end. Saturation. This implies that a transport molecule can only work so fast, and we only have so many. So let me try to illustrate saturation like this. Suppose that here's my cell membrane, and in that membrane I have a bunch of transporter proteins. So suppose that all of these that I'm drawing are transporter proteins. Whenever the concentration out here is modest, and the concentration in here is also modest, so we're going to make all of this active transporters. All of these are active transporters, and so they tend to want to grab these things when they get nearby and push them into the cell. And so it does that, and that one's gone. And so now there are 4 transporters and only 3 molecules. Anytime one of these molecules gets near one of these transporters, it's going to be grabbed and pushed in the cell. But what if we had hundreds of these molecules? Well, if this one gets down here near this transporter, and the transporter, it takes a certain amount of time to get that pumped into the cell. If this other molecule also moves over here nearby during that time when it's trying to move this one through, then it will just keep on moving along because this one is already active. If you have a certain concentration of molecules, eventually all of these will be 100% active or saturated, working at their maximum speed. Now let's take the kidney for example. So in the kidney tubules, you have something that filters, the blood filters in to this space, and there's sugar in this blood filtrate as it comes in. So you have sugar in the kidney tubule, where your body wants to reabsorb all of that sugar, and so it does. And typically a person's blood sugar is between 60 and 120, can be a little bit higher depending on how long ago they ate. But these transporters, they all are working pretty good, but once the blood glucose reaches 300 milligrams per deciliter, once it reaches 300, all of these things are saturated, and then you start to get sugar in the urine. So you'll start seeing all that sugar pour out into the urine. Now, in all actuality, you'll start seeing sugar in the urine before you get to 300, and the reason for that is because you have something called threshold. And threshold means that some of these transporters will get saturated periodically while others are not. So this transporter might not have reached saturation, but this transporter might be at saturation, or vice versa. And so whenever that starts to happen, you get to a threshold and you start to see some sugar in the urine, but all of the transporters become saturated at 300 milligrams per deciliter blood sugar. And lastly, let's talk about competition. This is the idea that molecules that are similar will compete with each other for a protein. In other words, let's suppose that I have a membrane here. Right now we're talking about transporters, but competition works for enzymes as well. So does specificity and so does saturation. All of these work for enzymes and transporters. So I have this transporter here. I only have one of them right here. And I have molecule A that looks like this. And I have molecule B that looks like this. This molecule right here is not the ideal molecule for this transporter, but it's so similar to the ideal molecule that sometimes it can trick the transporter and be pushed through. And since it's being pushed through, this guy is just going to keep on going down his merry way and get missed. He is competed off of the transporter. Now, an example of that would be like somebody who drinks methanol. Sometimes people get stupid and they drink things like printer ink that have methanol in them. Now, methanol by itself is not bad for you. It doesn't do anything to you, but your body uses an enzyme called alcohol dehydrogenase to convert methanol into formaldehyde. But there's another molecule that alcohol dehydrogenase likes to act on way more, and that molecule is ethanol, or we know this as just alcohol. It converts ethanol into metabolites, and ethanol has a much higher affinity for ADH. So if somebody drinks methanol, then you can actually use ethanol to intoxicate them, and it will bind to the alcohol dehydrogenase enzyme faster and more efficiently than methanol can. In other words, it will compete with methanol. And since the methanol isn't getting to the enzyme, you'll simply urinate it out. Now, just as something to keep in mind, I wrote alcohol dehydrogenase as ADH. Most of the time in physiology, ADH stands for antidiuretic hormone. And so I just want you to be clear that I am not talking about antidiuretic hormone when I talk about this alcohol dehydrogenase. Now let's go to the text and see what I've said. Molecules want to move into the cells. Waste needs to get out and nutrients need to get in, and some molecules can penetrate the membrane, some cannot. There are two primary routes into the cell. You can be carrier-mediated or non-carrier mediated. This is also simple diffusion. Most simple diffusion would be considered hydrophobic or lipid-soluble molecules. Sometimes some textbooks will consider channel proteins as simple diffusion. Channel proteins can be gated or non-gated. These include ion channels as well as water channels that are aquaporins. Carrier-mediated, you have facilitated diffusion and active transport. Now it's important to remember that anytime you have the word diffusion, diffusion means it's moving from high concentration to low concentration. Now, carrier and channel proteins have three important properties: specificity, competition, and saturation. And I won't go over what those are again. We just covered that, so we'll move on. There's something called passive transport, and there's something called active transport. And all these are just describing where the energy comes from. In passive transport, energy comes from the concentration gradient. Anytime I put brackets around something, that tends to mean concentration. And so since it comes from the concentration gradient, no metabolic energy is required. Active transport, on the other hand, requires metabolic energy. And so with passive transport, molecules are always moving from a high concentration to a low concentration. With active transport, you can actually move from a low concentration to a high concentration. So let me just go ahead and diagram this out for you really quick. If, let's say, these are your molecules, all tightly packed together, highly concentrated, and we say that molecules are randomly moving, they're vibrating, especially if they're in in a liquid or in an air, they're moving and vibrating in every which direction they want to. It could be any random direction. Now let's go ahead and zoom in on these molecules. So let's suppose that we have this molecule right here, and it is going to randomly move in this direction. What's going to happen? The first thing that's going to happen is going to bump into this guy, and then he's going to bounce the other way, and he's going to keep moving that way until he interacts with something else, and then move in another way. And so if there's a high concentration of a molecule, there are a higher probability of that molecule moving away from each other than towards each other, because once they move towards each other for a very finite amount of time, they will be forced to move the other way. And also you can think of it as the center guy. So let's go ahead and draw in a very central molecule. This molecule is at the center of all of these concentrations. You see that the only way for this guy to move is away from the center. In other words, the only way this molecule can move is if it reduces the concentration by moving away from the center. Okay, that seems fairly reasonable, easy to understand. And so the next thing I want to point out is, where does this diffusion stuff and active transport, where does all this stuff fit with what we just talked about? So let me go ahead and circle all of this, and let's make some more room. And we'll say that all the passive transport, simple diffusion, or it can be broken down into facilitated diffusion. Remember, if it has the word diffusion in it, it means it's moving from a high concentration, like we just talked about, to a low concentration. Now, what about active transport? So let's go ahead and talk about active transport. Where does it fit in the big scheme of things? What we didn't mention earlier is that there are two kinds of active transport. There's a primary active transport, and there's a secondary active. So with primary, ATP hydrolysis is directly responsible for ion movement. An example of this thing would be the calcium pump on the endoplasmic reticulum. So let me go ahead and show you what I mean. We have our cell, and inside the cell we have the endoplasmic reticulum. I'm drawing it fairly simplistically. And we have calcium inside, and I'm going to say that this is, say, for example, either a smooth muscle or a cardiac muscle, and we'll talk more about this later. But we have a ryanodine channel, ryanodine receptor, that when activated, calcium will just flow out from high concentrations in the endoplasmic reticulum to low concentrations in the cytoplasm. And then we have these calcium pumps that will pump the calcium back in from low concentration to high concentration. And so this requires ATP energy. The calcium pump is a great example of what we call uniport. Uniport meaning that you're pumping one object in one direction. Another example of primary active transport would be the sodium potassium ATPase pump. So what the sodium potassium ATPase pump is, here's my membrane, and here's extracellular, and here's intracellular. This is within my cytoplasm. And so I have this pump right here. And what you'll notice is that I have two potassium outside the cell. They're going to go into the cell, into this pump. Now remember, inside my cell I have tons and tons and tons of potassium, just absolutely chock full of it. But these sodium potassium pumps are going to get loaded onto the pump machinery. Now these sodium, this extracellular space is full of sodium, but 3 sodium just came off of this pump, and 2 potassium are going to load up onto the pump, and they are going to be moved back into the cell. So you'll see that they unload into the cell, and then 3 more sodiums get pumped out of the cell. And so basically the sodium potassium ATPase pump has 5-step mechanism. So first of all, 3 sodium move in, ATP gets converted into ADP, which the ADP will sit right here and block the exit. And then the ADP is released, causing a shape change to what we see over here. And that shape change allows the sodium to exit the pump, and it allows 2 potassium to move in. And then as soon as the 2 potassium move in, the phosphate is released, allowing the pump to move back to its original shape. We said that the calcium pump was uniport. This whole process is called antiport. Antiport meaning that 2 molecules are moving in opposite directions. It's also sometimes called exchange. This would be sodium-potassium exchange or sodium-potassium antiport. So now we have to go back to where we were originally. We said we covered primary active transport. Now we got to cover secondary active transport. This is also called coupled transport. Now, secondary active transport, the first step is ATP is used to create a concentration gradient. So we're going to pretend like we're talking about sodium and transport of glucose and sodium. We'll pretend like we're talking about the SGLT transporter in all of this case of secondary active transport. We have inside the cell, we have a low amount of sodium, but we're going to pump what little bit of sodium we have out of the cell using the sodium-potassium ATPase pump. That's primary active transport. So we're further increasing, and I'm going to make sodium red. We're further increasing our sodium concentration outside of the cell. And the reason we're doing that is because we want it to diffuse back into the cell. And so I'm going to draw my glucose concentration as blue. I have a very sparse glucose concentration outside. Inside the cell, I have a very high concentration of glucose. So to get glucose into the cell, it would have to move, and this specifically I'm talking about an enterocyte. So it works differently in other cells, but in the enterocyte, in order to get more glucose into the cell from the lumen of the GI tract, then I will use something called a sodium glucose co-transporter, which means that this is a channel that allows glucose to go in. It's a channel that allows sodium to go in, but it only allows sodium to go in when glucose moves in with it. So here again, I've redrawn my SGLT transporter. The key, the blue, is glucose, the red is sodium. Sodium, this is like a revolving door. Sodium should just be able to push right through it, but the door is locked. Without the key, without glucose coming in and plugging in here, the door won't open. But sodium uses its concentration gradient, it pulls in glucose with it, and it pushes glucose through into the cell. And so when we ask the question, what energy did glucose use to get into the cell, the answer is it used the energy of sodium's concentration gradient. But when we ask the question, where did the sodium concentration gradient come from, the answer to that is ATP. So I said the first step is ATP is used to create a concentration gradient. The second step is the molecule moves down its concentration gradient, pulling another molecule up its concentration gradient. And so the key is to remember that the energy for one molecule to move up its concentration gradient is obtained from another molecule moving down its concentration gradient. And I gave you the example of the SGLT transporter. So SGLT, and this is called co-transport, or we also call it symport. But there's another kind of transporter called the sodium-calcium exchanger, and there's also a sodium-hydrogen exchanger. And the sodium-hydrogen exchanger will cause sodium to move down its concentration gradient into a cell, and by doing that, it forces hydrogen to move out of the cell. So the way I kind of think of sodium, I think of sodium like it's the water moving under a big water wheel. If you remember those old movies where they have a water wheel and the water wheel is moved in a circle from water moving under it, and it powers energy for other things to happen. Sodium moving into the cell is like powering a water wheel that pumps another molecule either out in the case of hydrogen or in the case of glucose. We looked at energy requirement, and we said that we have an active transport and we have a passive transport. And so we called active transport primary or secondary. Now, I ask you, why do we not call the secondary, why do we not call it passive? That's because all active transport ceases if ATP is gone. So if you become hypoxic, you can't produce ATP for very long, and you would start to lose all of your active transport. On the other hand, if you have cyanide and hypoxia, both of these prevent ATP from being produced, and so you would have a ceasing of all active transport, whereas passive transport doesn't get its energy from that. It gets its energy from the concentration gradient, and so all passive transport ceases whenever the concentration gradient is equal. And so let's look at what I've said in the text. Energy requirement for molecular transport. Molecules always move down their concentration gradient from high to low. Okay, so when a molecule moves down its concentration gradient, no energy is required. It's called passive transport. When a molecule moves against its concentration gradient, in other words, it moves uphill, it requires ATP, and that's called active transport. Now, with passive transport, that can either happen through simple diffusion or facilitated diffusion. That means it is either lipid soluble with the hydrophobic effect, or sometimes we consider channel protein simple diffusion. It doesn't matter to me what you call it. Just do what your teacher, textbook, whatever says. It's simple terminology. But there's also facilitated diffusion, which requires a carrier protein. With active transport, you either have primary or secondary. Secondary, also called coupled transport. With primary, ATP hydrolysis is directly responsible for the movement of the particular molecule up its concentration gradient. For example, the calcium pump or the sodium-potassium ATPase pump. This one being uniport, this one being antiport. With secondary active transport or coupled transport, ATP first creates a concentration gradient of your secondary molecule so that the primary molecule can be moved uphill by diffusing the secondary molecule down its concentration gradient. The examples being SGLT, which is symport, and the sodium-calcium exchanger, which is antiport. And how do we know the difference between active and passive? If ATP is depleted, active transport stops. So hypoxia and cyanide poisoning will stop active transport. Passive transport only stops when the concentration gradients become equal. Okay, so we talked about moving into the cell. Now let's talk about molecules moving into the body or out of the body. And the way that happens is we call it absorption if it moves into the body from the intestine, or if something gets filtered into the kidney and the kidney decides, I want to keep that, and it brings it back into the body, we call that reabsorption. Okay, so what I've drawn here are two enterocytes, or GI cells. These are cells inside of your intestines, and they want to get molecules from out here down into your bloodstream. So this being your bloodstream right here. And the way you can do that is one of three ways, honestly. So you can either move the molecule into the apical membrane. So the top is called the apical surface. The bottom is called the basolateral surface. You can move the molecule through the apical membrane, through whatever means you have, and through the basolateral membrane, through whatever ways you have, and this is called transcellular. You'll notice I have two cells connected together, and it's connected together by a complex of things, and these are called junctional complexes. And the types, you have multiple types of junctional complexes. You have tight junctions, you have adherin junctions, and you have desmosomes. And so it's very difficult for a molecule to move between cells, but some small molecules have some ability to move through. Although this is not the primary mode, it does happen to some degree, and this is called paracellular. Now there is a third way for molecules to move through a cell, so I'll go ahead and draw the enterocyte again, and I'll draw a small invagination right here. You can maybe guess what's going to happen. So I had some molecule, some large molecule. It contacted the surface, and the surface started to invaginate. And the next thing you know is I have this whole thing pinched off inside of the cell. The membrane pinches itself off and traps within inside of it the molecule, and you'll guess what happens next. So this process is called endocytosis. And then next is the cell is going to spit it out at the bottom. So it spits out the molecule at the bottom. This process is called exocytosis. And so the whole process of endocytosing at the apical side and exocytosing at the basolateral side, this is called transcytosis. The thing you need to know about transcytosis is that it's for bulk transport and it's less specific. So lastly, let's take a quick look from the lumen of the gut into the blood. How does how does glucose get there? So we know that sodium first gets pumped out of the cell, or it is eaten with the diet. And sodium, using the glucose key, can push through the SGLT transporter into the cell. The glucose then builds up to high concentrations in the cell. After a while, it starts to diffuse out the GLUT transporter on the bottom, the basolateral side of the cell. Sodium, on the other hand, can either be repumped to the luminal side or repumped through the basolateral side through the sodium-potassium ATPase. So the clinical take-home here is water follows sodium. Sodium can actually move in fastest when glucose is present, and so if you want to quickly absorb water, you would drink it with something that has some sodium and some glucose in it. What does that sound like? And so the text says you need to move across the cell to get into the blood. So movement into the body. Transcellular, paracellular, or transcytosis. Transcellular, moving across the apical membrane and out the basolateral side. Paracellular, moving around the side. Very hard to do because of the junctional complexes. We talked about tight junctions, adherent junctions, and desmosomes. Now, two clinical applications. The one we just talked about, water follows sodium. Sodium is absorbed fastest with sugar. Fastest way to absorb water would be to combine it with a perfect ratio of some sort of salt and sugar. Now, another clinical application of what we just talked about is talking about how to get blood sugar low. Glucose moves through two types of transporters. We talked about the SGLT transporter, which is a secondary active transporter, and the GLUT transporter, which is a diffusion or passive transporter. So the GLUT transporter is what most of your cells have in your body. Whenever they are signaled by insulin, they move the GLUT transporter to the surface and allow sugar to diffuse into the cell. Most of our medications in the past have targeted increasing the expression of that transporter on the cell surface. On the other hand, within the renal system and within the GI system, the SGLT transporter is what takes care of glucose the most. So the SGL transporter will absorb glucose in the intestines. So if we block this transporter, we don't absorb glucose in the intestines. Rather, we feed the bacteria or we poop it out. And then the kidney, so the kidney will reabsorb the glucose in from the urine. So you automatically filter some glucose into the urine, and the transporter will reabsorb that back into the body. If we block that transporter, then we will urinate out all of our sugar. And like I've said here, the newer medications are aimed at blocking the SGLT transporter. Older medications wanted to increase the activity of GLUT. Newer medications want to destroy the activity of some of the SGLT transporters. There are several types of SGLT transporters, SGLT, and they're all numbered, SGLT1, SGLT2, and 3, and each different type is expressed in a different type of cell, and so you can specifically say, hey, I think I want to target the GI or the kidney. Now, the osmotic side effects are going to be pretty well known and easy to describe. So you're going to get diarrhea because you're going to have, if you block the absorption of glucose into the cell, you're not going to get water absorbed as easily. So you're going to have diarrhea, an osmotic diarrhea. And then you're going to get diuresis, which is also called urination. So it will pull water into the renal system because it's a solute. It's going to have an osmotic effect, and it's going to pull water into the renal system, and you're going to urinate more.